At the Oppenheimer Research Conference. I'd like to ask everyone to settle down before we get going. Before we get going, I'd like to bring attention to the illustrations happening on the right. The illustrations happening on the right are a summary or brief summary of each presentation. So during the breaks, feel free to walk over to Carlos just to get insights on how he summarized each presentation that's gone on today. Our first presentation for today is by Dabengwa, presenting on conservation insights drawn from millennium of grazing dynamics at HluhluweiMfolozi Park. May you please clap hands for Abraham?
Good morning. Okay, cool. Good morning, everyone. I'm going to be sharing some work that I did for a couple of years ago, but I've had time to think about it. So this is a way of how to do a whole ecosystem assessment using paleoecological proxies. Okay, cool. So I'll start off by looking at one of the biggest threats that we have at the moment, especially as wildlife ecologists, is what to do about the big animals. You know, the elephants, the rhinos, and also the large herds that we have running around the landscapes. Because the problem that they have is that when they get too many, sometimes they can degrade the environment. And some people prefer wooded forests compared to open savanna. And the other threat that we have is that these animals are also being poached, especially the rhinos, the elephants, and also there's this wildlife snaring that's been going on. While we're at that, we still have the problem of climate change. And one of the current solutions that we have is like, how do we trap carbon from the atmosphere? And forests have been proposed as one of the solutions. So you see immediately that large animals are actually competing for ecological space, for solutions, and also for resources with us as we go into the future. So today I'll be focusing on HluhluweiMfolozi Park, where we've got one of the healthiest surviving white rhino populations in the world. So in the map, you see at the bottom is the distribution of white rhinos in Southern Africa. We do have a population in East Africa, it's not shown in here. And then we also see the distribution of the protected areas around Africa. You see that some part of their range, they're actually not protected. And when we look further on the map on the right, we see that these animals are actually bulk feeders. They're in those areas where they are mostly grassy, and the resources that the animals usually eat are of low quality, but they're also viable for wildlife conservation. So let's take a bit of a step back and think about what's happened at HluhluweiMfolozi. So we've got these critical works that I just put out here. The first one just talks about the establishment of the park in 1895, the joining of the southern Umfolozi and the northern Hluhluwe, the causes of the proclamation of the park, which was extensive overhunting in the 1800s to the 1860s. It also talks about the documentation sources, how they tried to find out what animals were around in the landscapes, and also the interventions like veterinary that happened. Oops. And the second one is a critical book by Norman Owen Smith. I see you are here. It's about mega herbivore concept. Basically, these animals, these big animals, why are they important? It's because they can actually transform landscapes because of their body size, but they actually have got strong ecological connections such that we can call them ecosystem engineers or keystone species. And the most critical thing that came out of this book was the idea that in the late Pleistocene, like we can think about maybe, let's say roughly about 80,000 to about maybe 12,000 years ago, we lost a lot of animals, like especially herbivores, but in other continents, but in Africa, there's an idea that we actually retained a lot of our animals because of these mega herbivores that managed to hold on and provide critical habitats for other animals. And then, of course, there is the recent, well, it's not so recent, but this integration of the work that's happened in Hluhluwe, which talks about the consumer ecology, like the role of fire, the role of herbivores in terms of controlling the ecosystems. They talk about the critical thing, grazing lawns, which are these heavily grazed parts of the landscape which support the herbivores. And also they talk about the functional ecology. But we also know that in Hluhluwe, there's also a history of Iron Age of human occupation since like AD 400. So there's this critical work by Martin Hall. But what I want to talk about here is like the connection between grazing lawns and white rhinos. Because we do not have a long history of, are these grazing lawns recent, or did they just develop recently at Hluhluwe? For that, we have to go into the past. And then we already talked that in the past, there's been evidence of extinctions of herbivores. So the current crisis that we have now, some of them have got parallels that go in the past, where human beings have been involved. And one of the critical things that's lost during these extinctions is the resources. In this case, if you think about the grazing resources, they can get severely degraded. And there is a paper that came out suggesting that even in landscapes with heavily grazed landscapes with white rhinos, they actually cannot have an impact on the ecosystem. But can we find more? So paleoecological proxies can allow us to actually go back in time to, to use like for instance, with microscopic sporomyela, we can find out about grazing intensity or herbivore biomass. With phytoliths, we can look about like grass dynamics. Then we can look at the stable isotopes for carbon, nitrogen, phosphorus. We can look at soil organic carbon so that we can find out how much carbon is being stored in the system. And then we can also do other tests. So what I'm going to focus on today is this particular thing whereby we can actually use coprophilous fungal spores from dung to actually reconstruct herbivore densities. And how it works is that we've got a suite of coprophilous fungal spores. Some of them are specific to grazers or herbivores, and some of them are not. They're just generalist, but they all co-occur and can be used. So when we bring this information together, where we've got the specific and non-specific fungal spores, we just do some wrangling bits of mathematics, and we can actually come up with a grazing index. This is basically what we did at HluhluweiMfolozi. We collected a core, a 2-meter core. So on the board, like on the screen there, you do have SG, TG, and SG. So SG is short grass, which is your grazing lawn. TG is tall grasses. So we collected the sedimentary sequence where we actually looked at these boards. And I know there's a lot of results there, but what I want you to focus on is this one. The first one shows that after 1,700 BP, soil organic carbon declined. Next, we see that during that time of decline, carbon-nitrogen ratio was low, which means that the food might have been palatable. Critically, we see that soil erosion increased after that period. I think this is like 1,580. And then we see that fire decreased in that period. And so we infer here that the top section is a grazing lawn, the bottom is like grassland. This just shows a overview of the coprophilous fungal spores, the specific ones and the non-specific ones. What I want you to look at is that sporomyela on the red line going up was increasing, whereas Coniucita lignaria, which is mostly to do with soil moisture, is going down. Let's get more specific. We see here in that graph on the left with the rhino and a buffalo, it's showing that actually if you relate grazing pressure that we've got from the spores with the sporomyela, which can just give grazing biomass, we can find a bimodal distribution, which tells us that you've got the system can be in a level where it's got maybe low grazer biomass and then high grazer biomass. So those are two stable states, and we know that rhinos are maintaining the one with high grazer biomass. And we see that high grazer mass with the brown line goes steadily up over time. But we see that soil organic carbon in the green and the black arrow, which is like fire, is actually going down. So we see that heavy grazing actually over time reduces vegetation biomass, maybe through roots, and also above and below ground, which increases carbon in the system. So we see that large grazers can actually deplete carbon in the system. And for here, it's just showing the relationship within the bulk density and the grazing pressure. We see that it's a nonlinear relationship, but it's an inverse relationship with soil organic carbon. There's a lot of studies that have looked at this, but I will not get into it because we already know that herbivores, we already linked that with soil erosion up at the top. So the first thing that you see at the top, you see the rainfall, and then at the bottom you see the soil organic carbon. We can already see that rainfall is definitely driving the system. But then we also have information from Martin Hall's thesis that actually pastoralists arrive in this region. Okay, so let's put this together. So, as you can see, we've got the system at the top where it could have had low to intermediate grazing intensity. This could probably be in animals not spending much time in the place. And then it shifted because of drought and fire to a system whereby you were maintaining a high grazer biomass and heavy grazing. We already know that white rhinos are the ones that actually have been persistently linked with heavy grazing and HluhluweiMfolozi. And this study shows that actually that these grazing lawns are not young, they're actually quite old. They're older than we think. Secondly, we see that the grazing lawns are resilient because they've persisted over time. The same place that we went to core, actually those grazing lawns are much older, which has actually been hypothesized for other ones in East Africa. Although we are uncertain about, because one of the things with fungal spores is that we cannot differentiate between domestic grazers and wild ones, so it's still a bit iffy. So at the moment, this is where we are. But we already see that climate change, we saw that climate drives the system. We already see that this is actually valuable information for someone wanting to conserve ecosystems into the future. So we want to get to work with more people, conservationists like parks, researchers, to get this work going forward, because this is just one site, a local site. We know that for meaningful progress, you need to establish this network of sites to get these key critical insights. Secondly, these connected sites can allow us to look at the scale level effects of these processes. Because in China, they've shown that grazing intensity can be linked with the spores can be used for assessing grazing intensity, but they are basically looking at herbivores that are domestic. So we need to build models which are relevant. So at the moment, we're working on this. And then we also want to integrate novel approaches like sedimentary DNA to find out, are these domestic or wild herbivores? What are they? Are they mega herbivores or meso herbivores? With that, we'll get into, as more data becomes available, we'll get into machine learning. And finally, we hope that this can be used to guide meaningful conservation policies going into the future. So I'd like to thank people who have been critical in this work, my previous supervisors, a lot of our sponsors, and also thank you for giving me time to share my work.
Thank you, Abram. Before you get off the stage, we have time for one quick question. Does anybody have a question? Second around. Lucky. Up next, we have Belinda Reyers talking on bending the curve of biodiversity. What are we trying to change? Thank you, Belinda.
Morning, everyone. It's really lovely to be here today. Duncan, we were trying to remember when last we saw each other, and I think it might have been at one of the first of these conferences, but it's hard to remember that long ago. So thank you to you and to Rendani and your team and to the Oppenheimer family for the invitation and support to be here. So I'm going to be giving a slightly different talk, not a conventional research talk, but a talk that presents recent results that were highlighted by, oh, sorry, I see my timer hasn't started, so I'm just going to start my own timer. Give me a second. There we go. I don't want to blather on for too long. Any case, so there are these two big science policy practice assessments that have been published earlier this year, in which I was involved, that I think hold some interesting food for thought in terms of answering this question that's been bothering me over the last couple of years, which is this question of, in terms of biodiversity loss, what are we really trying to change? And the context behind these assessments and reviews, and also behind this question, is something I refer to as conservation's conundrum. Despite increasing and concerted efforts to conserve biodiversity, shown there on the left-hand panel in green, protected area coverage, funding for conservation, and national biodiversity strategies and action plans, we still continue to see worrying trends in biodiversity, which are actually accelerating in many cases, as shown there in the right, with the Living Planet Index, the Red List Index, and the Biodiversity Intactness Index. And this conundrum, fortunately, or perhaps unfortunately, is not only ours. There are many other sectors in society struggling with a similar conundrum across other ecosystem and Earth system variables, but also across important societal well-being variables. For example, here we see the Human Development Index, which is a combination of education, life expectancy, and another variable I can't remember, where we saw a big crash during COVID, but actually a permanent shift. Up to 50% of developing countries are not on a path of recovery. So we're now proposing that there is a permanent shift in human development ongoing. Income inequality, we saw some interesting gains in terms of countries becoming more equal with one another, but that's quickly been overtaken by the poor and the rich within a country becoming less equal with each other. So we're seeing within country inequality beginning to rise. And many might say, yes, this is the problem of developing countries. But actually, if you look at that graph of OECD learning outcomes, we're seeing it across a wide range of countries, across maths, across all kinds of important learning outcomes. We're seeing worrying declines in education variables, too. And I think this has helped us to realize that we're not dealing with an isolated crisis. The biodiversity crisis is part of a complicated and complex set of interconnected crises, which has now won the label of a polycrisis, where we see geopolitical crises affecting social instability and having feedbacks from ecological declines and crisis. And I think this realization that biodiversity loss sits in the center of a very complex web of change and flux and uncertainty has prompted many of us to sort of take a step back, to zoom out, and to really ask some questions about the system that biodiversity loss is located in, and the conditions for the system-wide change or the fundamental rewiring of these systems, of energy systems, of economic systems, of justice systems, and of conservation systems to be able to deal with these interconnected crises and to work together. But before we become paralyzed by the multitude of interconnections that actually affect the work we do, fortunately for us, systems thinking and system approaches offer a lot of tools and methods which allow us to take advantage of these interconnections. Instead of zooming out, there's an opportunity to zoom deep. And this is one of the tools, a system change tree. It's often depicted as an iceberg, but I thought a tree would make more sense to an audience who hasn't probably seen an iceberg. And this tree encourages us to think below the symptoms. So the symptom of biodiversity loss, to start digging deeper and think about the relationships and structures that underpin this biodiversity loss, and to go even deeper to think about mindsets and heart sets that lie and are interconnected across these systems. And importantly, what we're learning is these deeper points actually have greater leverage. So small changes below surface can actually cause large changes that ripple around systems. And so using the systems tree or systems thinking approach to change, I really wanted to examine, what are we doing to move away just from this idea of managing the decline of biodiversity to, as Georgina Mace pointed out, to be more bold and to actually think of actions that can restore the abundance of nature to levels where both people and nature thrive. And this is a wonderful graphic that shows this idea of business as usual in gray, in yellow, what we're trying to do in terms of increased conservation efforts, and this green sort of hypothetical idea of not only making things less worse, but actually making things better. And of course, everyone has a hypothesis of what should be there where the green question marks are, and I'm going to advance some ideas of what those question marks might look like. So, in terms of thinking about that system tree and biodiversity loss, we know we have a lot of frameworks in the conservation sector for thinking about what causes biodiversity loss. And all of us are very familiar with the 5 main drivers of biodiversity loss: the land use change, the invasive alien species, pollution, etc. But I would argue those are also still symptoms, right? They sit above the surface of the real problems. And so some of us have ventured below this and started to think about economic systems and technology systems and trade systems and how those are perpetuating many of those drivers. And these have been labeled the indirect drivers. But more recently, people have thought, well, what actually is lying beneath these systems, the way the world and society are structured today? What are the underlying causes, the paradigms, the value systems, and the beliefs that have given us the world we have today and all the symptoms of crisis that exist? And so fortunately, this is one of the reviews. The Intergovernmental Science Policy Platform on Biodiversity and Ecosystem Services, which sort of rolls off the tongue, recently released their assessment of the underlying causes of biodiversity loss and determinants of transformative change. And in this, they find good evidence from a thorough review of the literature and of various sources that they found at least 3. The first is actually the one that Nikki Oppenheimer mentioned this morning, this disconnection and domination of nature and people. The second one was highlighted as the uneven distribution of power and wealth around the world. And then the third one is the prioritization of short-term individual and material gains, have been highlighted at this stage as the 3 main underlying causes of biodiversity loss. I'm only going to focus on the first one. At this stage, it falls more in my area of expertise and is one I think that might resonate with many of you here, because we're all worried about this increasing disconnection from nature. We're seeing it making newspaper headlines, a 60% decline in 200 years, says The Guardian. It's not just kids. There's that study at the top which shows American adults, more than 50% of adults, spending less than a few hours each week outdoors. A really worrying trend. And then a study I really like is that graphic presented there, which shows the decline in references to nature in song lyrics over time. And we see the same in fiction, in books, in movies, in TV series, less and less are representing nature and the outdoors. And so really a sign that our art and our culture and our society are becoming increasingly disconnected. Oh, there's the timer. Sorry, it moved. And so we're all going to say, well, why is this? And there's the simple answer. Those are the reasons for the disconnection, as summarized in a recent paper. I think important to recognize in this is that there are individual reasons and societal reasons. And so I think that is very important for us to recognize as a community. It isn't about individual change only. There's big societal changes required as well. But instead of going through what we need to do to get others to connect to nature, I thought I wanted to look at our sector. And this is the second review that I'm going to talk about, which is what does this disconnection look like in conservation science and practice? And fortunately for us, the late and very much missed Dame Professor Georgina Mace did this review in 2014 where she really asked this question of, we know that conservation has shifted over the decades in terms of its framing and its purpose, but she was asking the question of how has this caused us to change our perspective on the relationships between people and nature and that connection. And this is the framework that she came up with, showing the changes in conservation, global conservation science, from the 1960s to 2020, where she looks at how first conservation was conceived as nature for itself, the idea of wilderness and protected areas, and species conservation. This then changed in the 1980s as we see books like Silent Spring and the concern around pollution and the threats of humans to this idea of nature despite people, where we see a rising interest in extinctions and threats and threatened species and overexploitation. We then see another shift in the 1990s and early 2000s to this idea of ecosystem services, the fact that people need nature for provisioning, for goods, for culture, and for spiritual and recreational reasons. And she called this the nature for people. And then she proposed that there was this emerging framing that she was seeing in 2014 called the people and nature, where more and more our sector was starting to recognize that we need to be studying both, we need to be working across the disciplines, and we need to be bridging the social and the ecological sciences. And so I've just done a very simple depiction of how she summarizes what those views see for the relationship between people and nature. And so the first one, obviously, there's no people in that framing of conservation. The second one is people are there, but they are seen as the problem. The third one, we see nature and people are connected, but very much in an anthropocentric, natural resources, nature for people. And then the last one that she proposed really is interested in those interconnections and flows between nature and people. And I must say, these last 10 years, we've seen more and more of this framing emerging in the sector and in the science and in policy. And we've seen a very productive and effective conversation around people, nature, connections, new courses, new PhDs, new university schools, all bridging those disciplines and really starting to think about the bidirectional flows connecting people and nature. But interestingly, across these depictions, we're quite challenged by the fact that people and nature are still seen as separate and separable, right? There can still be circles around each. This is nature, this is people, and this is the arrows that connect them. And I think, a lot of our conservation training, a lot of the textbooks, a lot of the policy discussions, at the back of our minds is this paradigm that nature and people are separate, and even as Nikki Oppenheim was saying, man is dominant, that hierarchy there shows. And so we draw circles and say this is included and this is excluded, and it filters into all aspects of life. If you look at that graphic of splitting the natural and social sciences, it affects what we do at school, it affects what we study at universities, and it affects then the sectors that we go in to work. And this graphic actually comes from a study that was done of children that noted that up until the age of 5, children don't cluster knowledge into natural and societal. They cluster it in many different ways. And it's only as we start entering into formal education programs that we actually start to see the two as separate and separable. And I think this is quite problematic. Many of us are challenged by this fact of pitting society against nature. It leads to a lot of basic assumptions like this horrible Kuznets curve that many of us were trained with at university, that countries must develop first, and when they're rich enough, they can look after their environment. And that has led many of the conversations around GDP, around development, environment as luxury. So many meetings I've been to where I stand up and say I work in environmental sciences, and I'm told, oh, you're anti-development or you're anti-poverty. It really is a terrible false choice, I think, and dichotomy. And here's a graphic from a recent publication. On the x-axis, we've got ecological footprint, and on the y-axis, we've got social goals being met. So ideally, you want to be top left, where that little green circle is. Social goals are all met in a minimum environmental footprint, or a footprint that amounts to the amount of planet Earth that we have. And no one's there. Lots of people have developed very well by using the environment, but not many of them have then gone back and used that development to plow back into the environment. And in fact, there's little old South Africa sitting in the middle left, where we've been quite heavy in terms of our environmental footprint and haven't really made great progress in many of these social targets as well. And then we see the countries on the far right who have had a disproportionate effect on the environment to reach most, but not all, of their social goals. So this false choice, this idea of a Kuznets curve about the fact that we'll care about the environment later, doesn't really seem to be manifesting in the evidence. And so we're left with this concerning picture. Here are the 17 Sustainable Development Goals. The green ones are targets that it looks like we'll meet by 2030. The yellow and the red, we won't. So across all measures of environment, ecosystems, and society, we're falling short. There are, of course, alternative perspectives on people, nature, separability, and relationships. We know, for example, many indigenous peoples and local communities have a far greater reciprocity and relationship and perspective of their inseparability from nature. We also know many fields of social science don't study the two as separate and focus more on concepts such as relationality and relational resilience. We saw a great example in Gladys's presentation about how you can't draw a line between humans and nature when it comes to disease transmission. And similarly, our gut microbiome is nature, and it lives within us. So many of these are starting to elaborate on this challenge of the separation and the artificial and arbitrariness of drawing lines between the two. Now, I've been very lucky to have spent a lot of time with the Stockholm Resilience Center group in Sweden, where they have really been developing this idea of social ecological systems, where they've emphasized that we know that complex systems and their behaviors and properties are defined far more by their interactions between the parts than by the parts themselves. And this is that whole, the idiom that many of you are familiar with, that the whole is not the same as the sum of the parts. So really thinking about how we study systems as whole systems rather than trying to break them up in reductionist ways. The same intergovernmental panel has now done a wonderful review of all these alternative perspectives and is working hard on making sure that they come up in policy, so we see a greater connection between nature and people in the biodiversity policy arena. It is, however, important to note that some of the less anthropocentric are still rather marginalized and not necessarily much included in some of these conversations. And so we tend to see a dominant, the ideas of natural resources, of ecosystem services, and landscapes coming into these conversations. So here is that last piece of work. A colleague and I then went to go and revisit Georgina Mace's framework 10 years on to say, are we seeing more of these marginalized perspectives coming through in conservation science? And the answer is fortunately yes, as we see knowledge being mobilized in science from indigenous communities, from the global south, and from social sciences. We're seeing far more acknowledgement of these intertwined relationships between people and nature. And we've now proposed that we've moved into this fifth perspective, which we've called people with nature, a more collaborative and less competitive idea where we highlight the inextricably intertwined nature of people and nature, and really the need to shift our focus away from trying to understand the parts to trying to understand what connects the parts. I think, interestingly, in this perspective of people with nature, we see a much broader engagement with the disciplines. We see us moving out of university disciplines, out of just engagement with the social sciences, to engage with practitioner knowledge and policy knowledge as well. We also see a nice departure from this tension between the intrinsic values of nature, the fact that nature has value in and of itself, and the instrumental values of nature, that nature has value because we need it, to a new value system, which is more the relational value system. And so I think we're seeing a broadening and a diversifying and a greater inclusion in this final frame. And I think it's really important to know, as we do in this review, that all of these perspectives exist at the same time. In this room, we're all thinking from different perspectives in terms of that relationship between people and nature. And I think this argues for a greater flexibility and a greater awareness in our community to be able to deal with these multiple perspectives and to make sure we're not marginalizing those that see a deeper connection between humans and nature. I'm just going to finish then with a couple of advances and exciting areas and avenues that I see in this people with nature framing, which I think might be relevant to some of the conversations coming up. So the first one is we see a growing evidence base of how people with nature is actually fostering reversals in the loss of biodiversity. So this study on the left is from Amazonian carbon loss and differentiates in the first column between indigenous territories, the second column protected areas, and the fourth column are other lands. And we see how forest conversion is negligible in indigenous territories, and we see increasing levels of degradation and disturbance in those lands, but nothing compared to what we see in other areas. Similarly, we've seen this interesting study published, which looked at France and Colombia and studied and quantified human-nature connectedness, and then studied the correlation with pro-environmental behaviors like volunteering or picking up litter or donating money. And the answer is quite obvious to many of us. The more we see that connectedness between humans and nature, the more pro-environmental behaviors become. And they found interesting results around age and the effect that age has on this, as well as distance from urban centers and how that affects our perceptions and experiences. There have been some really nice advances in terms of new approaches that are available to adopt this more intertwined and relational perspective. Biocultural diversity and biocultural approaches have been around for quite a while. Here's a nice review that was done a few years ago that shows how they're really picking up. And this is wonderful work that really emphasizes humans and culture and how those are intertwined with nature. A lot of these studies are more on the descriptive side than on the applied side, but I see a lot of potential here for moving this into application. And then right here in South Africa, we have the wonderful work of Jessica Coburn and her group, who are looking at how we can focus more strongly on relationships at a landscape level in conservation. And she has now depicted and piloted and tested this more relational approach to landscape stewardship across 6 different sites, I think, in South Africa. So lots of nice, exciting advances and approaches available. But I think Nikki Oppenheimer raised this point as well. We live in the 21st century. We're not all rural, we're not all local. We're very cross-scale, we're very connected. There is no local, isolated area on the world that doesn't get impacted by the changes. We're hyper-connected, and we're rapidly urbanizing. This graph shows that the urban population has overtaken the rural population and will continue to do so. So what does human-nature connectedness, relational approaches, biocultural diversity, landscape approaches, what do they look like in this hyper-connected and urbanizing world? And I think this is a real challenge for us as a sector and a society, is to start answering and taking on some of these challenges of applying these approaches and adapting them to these kinds of contexts. And I'm going to end off with some opportunities that I see. It's very easy in these kinds of talks to get quite depressed about the hopelessness of the challenges ahead. But I feel far more enabled and empowered to do my job now that I have partners that I'm not in competition with. For so many years, I was the sole voice saying, but what about nature? What about conservation? And was being beaten down because we had to first solve poverty and food insecurity. But now we're actually seeing interesting collaborations because of this recognition that society and planetary imbalances and crises are linked. And so here's just a slide of some of the novel collaborations that I've been part of or seeing coming up, both because they're addressing the same sort of challenges, poverty or food insecurity or gender inequality may not sound the same as biodiversity loss, but often if you go to those underlying causes, we're struggling with the same root causes driving our systems in these directions. So here are just some examples of projects that we see biodiversity and other sectors coming together. And I'm just going to end with this sort of more hopeful message, which actually doesn't come from our sector, but comes from the human development sector, which really raises the hopefulness in thinking about it's not about trying to get back what we have or restore to what it was like before. We're a different planet. There are 9 billion people. We have unique challenges. We're all very interconnected. But we really do need to rethink and reimagine our futures, renew and adapt the way we work and the stories we tell about the work we do and what we value. And I think this holds a lot of hope for much of the work that I'm excited to hear about for the rest of the conference. Thanks very much.
Thank you, Belinda. We can take a few questions. Do we have any questions? Duncan.
Belinda, thanks so much for a great presentation. I was just interested with this new sand parks in a Vision 2040, what your views are, because I think that's going to change the conventional view on conservation a lot. And obviously with the South African perspective, that there will be challenges, but yeah. I think there's an exciting opportunity which links into what you've just said.
Yeah, I do think, South Africa is actually at the cutting edge of a lot of this more collaborative, more integrative way of working of people in landscapes with nature. And Sandparks has been championing a lot of that as well. And I think in these, are you referring to the protected area expansion? Yeah, I think that we're going to move ahead seeing protected areas being conceptualized in very different ways from the nature without people or the nature despite people kind of ways that they were originally set up. So I agree, loads of challenges, but also I do think this is sort of an innovation hub going on in Southern Africa for these kinds of new approaches.
Thank you, Belinda. We can take one more question. Do we have any other questions? Ricky?
Thank you, Belinda, for a very inspiring and great comprehensive review speech. Question, what is your sense of the opportunities we're facing, considering that the most powerful country in the world, the United States, the federal government has turned their back on global environmental and social development issues, and actually in the European Union, the reduction of to biodiversity environmental funding is more than 50%, as I understand, over the past 12 months. What are the opportunities for us sitting here in Africa?
Well, thank you, Duan, for that very easy question. I'm happy to discuss it more with a drink in my hand, because I think it requires alcohol. I mean, I think we're seeing a lot of examples being set of what not to do. I showed that graphic of conservation funding, and actually over the last 2 years, we've seen it start to reverse on that. But we're also seeing conservation financing picking up, climate financing picking up. So I'm not sure I see opportunities in those two examples other than to not go there. But I do think there's an opportunity on this continent and through collaborations that exist within the Global South to carve a different path and to learn from that graphic I showed that development first and environment later is a recipe for disaster and leads to a polarized and unhappy society with political tensions as well. That escapes your question, I think, nicely, right?
Thank you, Belinda. Up next, we have Merlyn Nkomo speaking on navigating the green dilemma.
Hi, everyone. As has been said, my name is Merlyn Nkomo. I will be talking to you about the green dilemma. This is some work I've been doing for my PhD. But as a reflection, this is my third ORC. The first one, I was just an undergraduate, and now I'm presenting my PhD work. A lot of growth, and I'm grateful for. So we've been told about the climate crisis by prophets of doom, but what happens when something that is desirable, like climate action, is at odds with biodiversity conservation? Sorry. So South Africa is leading the charge in Africa in terms of decarbonizing the economy. We've got about 34 wind farms producing upwards of 3.5 gigawatts of energy. And unfortunately, renewable energy is contributing quite less than we would hope for. Wind energy is producing 9%, as opposed to coal, that is over 80% still. We're planning and trying to work towards that being up to 25% by 2030. That is a tall ask, but at present, we have about 21 projects that have been approved, and those 21 contribute about 2.5. And there's great investment to get us to over 69 to 76 gigawatts of energy. This is a huge jump in a short space of time. It may look crazy and scary to conservationists, but if you look at the developed world, countries like Germany are already producing that much, followed by the UK, Spain, and France. And in comparison to South Africa, they do not have as much land area, they do not have as much coastline. So South Africa is actually in line to producing its wind potential and growing its economy. But unlike these countries, South Africa is a very important country for biodiversity conservation, with biodiversity hotspots of its own, high rates of endemism, and very rich avifaunal species richness and unique biomes within its borders. Also, as a raptor biologist, what I care about the most is that South Africa is an important place for raptor research and conservation. There's priority being put to learning more about our raptors here. We've got high diversity in raptor species, but very low research output compared to the developed world. So that's where I come in. I'm working with this very handsome bird, the jackal buzzard. It is a medium, near-endemic raptor. It ranges in southern Africa with the core of its population within South Africa. However, it is the most killed bird by wind turbines. Let me correct that. The most killed raptor by wind turbines. It constitutes about 10% of all mortalities and 22% of raptors. The problem then is that it is of least concern, so it takes second place to species such as Cape vultures, black harriers, which would be next year, the black harrier, which will be next year's bird of the year, Vero's eagles, Marshall eagles, more charming species. But if you look at the rates, which are quite conservative, the rates of mortality, it is quite high up there as the most killed bird by wind turbines. So there's also been work that has had a species focus on those big birds I showed. There's been production of guidelines for development and mitigation for Vero's eagles, Cape vultures, and black harriers. There's work being done on Marshall eagles at the moment, and a lot of research outputs specifically talking to the nexus of wind development and these species. But none has been done on jackal buzzards. We don't even know the population of jackal buzzards or really any basic ecological knowledge about this very common species. So for my work, I started with stakeholder engagement, talking to stakeholders in the landscape, policymakers, avifaunal specialists in the wind sector, developers and researchers and conservationists about what should we focus on to better conserve this species. So I set out to ask them about their opinions on the adequacy of EIAs for this species, the importance of certain threats to this species, the frequency of collisions if they knew about it, the scale of impact on the population, appropriateness of interventions, and what they thought priority areas for research would be. What we found was that most stakeholders had faith in the current EIA methods. They thought that direct collisions compared to habitat loss, electrocutions, and other disturbances that come with development were the most important for the species. They didn't think, however, that it would be likely that these impacts would translate from the local populations to the national population of jackal buzzards. But there was really uncertainty of the scale because we don't know the population. However, there was willingness to apply mitigation and prioritize jackal buzzards if avifaunal specialists and their outputs in terms of EIAs prioritized them for mitigation. So we set out to do some population modeling that would show that we might be going towards a trend where we would need to act on this quickly, sooner rather than later. So we wanted to show the cumulative impacts of turbine collisions on this very common species. So in South Africa right now, we have very rigorous EIA methods, and just the sector in renewable energy is very cooperative. They work as a community, and they're very cooperative. They're doing a lot more than a lot of industries are doing. What they do, however, in terms of how to mitigate for impacts is they have an EIA done by avifaunal specialists, which demarcates what priority species are, and then also what target species would be. Target species are not, priority species are not always target species, and target species are the ones included in adaptive management and prioritization for mitigation. Jackal buzzards, because they're not of least concern and they're abundant, unfortunately, are not always prioritized as target species. So we had scenarios of development in which we could double the number of wind turbines we have in the country, quadruple, sixfold, and tenfold. And we modeled this on Vortex to see management thresholds that align to a over 30% decline according to the IUCN in 3 generations length. That would mean the species becomes vulnerable, or 50%, that would mean the species becomes endangered, or over 80%, that would mean the species would become critically endangered. We had a crude method of estimating population size using suburb 2 pentads and resightings of the birds, reporting rates of the birds. And we had a range of population estimates. A low one, which would have individuals just above 16,300, a medium range one, which would be about 32,000, and a high estimate, which was over 44,000. These were crude estimates. What we found was that on the lower end, the birds would really be in trouble because in 3 generations, which for this species would be 26 years, all the development scenarios meant that they could become vulnerable, endangered, and critically endangered. The same applies for the mid-range estimate. Please keep in mind that these are not real estimates. These are a demonstration of that it is crucial for us to know the starting population of the species. We also added a scenario where there would be 50% mitigation or 50% reduction in the fatalities, and you see in those dotted lines that it quite makes a difference that we start to prioritize the species for mitigation. I'm going to just highlight quickly work that is being done by other colleagues, led by Dr. Rob Simmons with Birds and Bats Unlimited. They piloted the blade patterning, and in 2022 they got permission from the Civil Aviation Authority to mark our turbines with signal red stripes patterning. And this trial was first done at Umoya on just 4 turbines. There's a lot to consider when this is being done. They were fortunate enough that the producer of these turbines, Vestas, was willing to maintain warranty on these painted turbines. This means halting a lot of production time and very, very high costs in patterning these turbines. They had aimed for 10, and they could only, in the end, afford 4 turbines. But their results are really encouraging. You'll see that the patterned blades showed up to 87% reduction in deaths of raptors. There's a lot of other markers for different species, but there was visible and encouraging results, and a lot of wind energy facilities are willing to try this and work out warranty and production. This is a passive mitigation method, and it could do a lot of good for different species and hopefully jackal buzzards. So in closing, my work shows that wind energy developments, if we're not working together as conservationists, can even lead to locally and common species declining. And that jackal buzzards, as common and abundant as they are, could also decline to extinction if they're not prioritized for mitigation. It also shows, we also found that policymakers usually were not in the know about critical information like certain species, and they need to, like jackal buzzards, when they need to consider this in their permitting of development sites. Stakeholder engagement and adaptive management is going to be very important because we had to work with a lot of stakeholders, even developers across the seas who have to agree to waive a warranty for some actions that we're trying to see how they impact species. So I thank you. This is where I'll stop today, and I'll be happy to receive any questions.
Thank you, Merlyn. Do we have any questions for Merlyn? No questions?
Thank you.
Thank you, Merlyn. Well done. Up next, we have Eduard Venter speaking on the soil fungal diversity of Tswalu Kalahari Nature Reserve.
Good morning, everyone, and thank you very much for inviting us to present some of our research that we're doing in Tswalu Nature Reserve. Obviously, this is not work that I'm doing by myself. We have quite a few collaborators at the University of Pretoria, as well as some collaborators from Australia. You all know fungi, most probably as that green furry thing at the back of your fridge, or something that you have with your braai or in your pasta, but obviously, they are quite a big part of our ecosystems. For instance, the biggest problem that we face is with food spoilage. And as you can see on the maize, it is quite a huge problem that we face. If we look at the bottom there, you can see also quite a bit of human medicine is produced from this. For instance, antibiotics that we all know. But that specific instance is Parkinson's medication. And then some of the other things is biopesticides. This is quite a huge market, especially in the European Union, because of chemical bans. So all in all, it makes up a huge part of our lives. What we're currently studying is the fungal biodiversity for South Africa. And the problem that we face here is that a lot of work is being done for agriculture, but not specifically just to determine the baseline of fungal spread within South Africa itself. So to this end, we've started with the grassland biome, because that's where 90% of the agricultural activities takes place. And we've looked at all of the nature reserves that are found there, because we're interested in soils with low anthropogenic disturbance. We've expanded that to the Kalahari and the Karoo, but specifically in the Kalahari, we're focused on Tswalu Nature Reserve. The techniques that we're using is, the zoologist calls it environmental DNA, but we've been calling it a longer time as metabarcoding and metagenomics, which we try to establish what's the total spread or distribution of fungi or microbes within soil. We're using both these high-tech throughput technologies, but also classical isolation, because we're interested in the specific genus, the Fusarium, or genus Fusarium, which is one of the biggest plant pathogens in the world. They also produce mycotoxins, which affect us. For instance, it causes esophageal cancer. Oh, sorry, that's fine. OK. When we went through Tswalu, we were really interested to get in-depth sampling. So we sampled from a north-south as well as a east-west, and we obtained about 50 sampling points. The samplings that we do is on a 15-meter transect. Maybe if I add the arrow, that will show it a bit better, where we sample at the tops and the two sides of the arrows, as well as in the center. So we get a composite sample of about 5 samples per 15 square meters. These are all put into one bag, and we take that back to the lab to do extractions. We extract total DNA from the soil, convert that into library, and that is then sequenced. And in our case, this was through the Illumina system. And that generates quite a lot of reads that we then process to get information that we can analyze further. So looking at the phyla that occurs within Tswalu, if you look at that image, it's dominated by the red, which is the Ascomycotina. Those are the ones that you usually see in your fridge or in fruit that they colonize. But you'll also see lesions on plant leaves quite extensively. And that's about the macrostructures that they make. In yellow, the second largest group, those are the basidiomycetes. These are the mushrooms, bracket fungi that you see on trees, but also rusts, which is the predominant problem in South Africa for wheat production. These are obligate pathogens of cereals, for instance. And then there's a couple of other ones that I'm not going to talk to you about today. So how does the Tswalu data fit in with the grassland work that we've done? I must say I was a bit biased, so I suspected that we will see less species or isolates here in the semi-arid regions than what we found for the grassland. But much to my surprise, we identified much more. We have about 13,300 isolates from Tswalu, compared to about 9,500. But take in mind, this is across the whole grassland biome of South Africa. So Tswalu's richness is much higher than that whole largest biome that we have. If we further look at the data, excuse me, the data is analyzed against databases, and unfortunately, the database has a lot of unknowns in them. And if we look at the Tswalu data, you can see about 25% of the data we generate is unknown. We don't know what they are, we just know they are fungi, and up to about family level. For the grassland isolate, that was about 18.8%. So even there's this discrepancy. Semi-arid regions have a lot more unknowns than what we find for grasslands. But it sort of stands to reason, because grasslands have been studied a lot more. People either focus on something they want to study because of its economical value, or something that's very extreme because of its nature. It's interesting to find out what is occurring there. If we focus a bit more on the biodiversity within these samples, you can see that we have very high biodiversity at each sampling site, with the majority of our index showing that it's above 0.9 or 90%, meaning that the diversity is very high, except for a couple of samples that you can see there. If I go back to the map and I show you where these sampling sites occur, you can see that there's 5 of them, roughly, but 2 of those are very interesting, which I forgot to mention on the first graph that I showed. The one of those is dominated by ascomycetes. So not a lot of other diversity. Everything we find in there, or probably about 95% of the samples that we got back, comes from the ascomycetina. The other one there on the left was about a 50-50 split between Basidiomycetina and the ascomycetina, which is also very interesting, because if you think back to that slide, the Basidiomycetina was not that a dominant presence within any of these samples. So that's really an interesting observation, which we're looking into a bit more. But what I would like to focus on, we've got quite a few interesting fungi, but I thought this one would be interesting to just have a quick look. Kalahari truffle, or the genus Kalahari truffle, has only one species in it, Phalliae, and it occurs quite extensively in the Kalahari, hence the name Kalahari truffle. But if you look at the distribution throughout Tswalu, you can see it is quite extensively found. It's not the most dominant species, obviously, that we found, and that would explain some of the discrepancies where you see some sampling sites don't have the specific isolate represented. So this speaks to some of the limits of the techniques on sensitivity. Depending on what technique you use and what specific region that you focus on, you will get better and worse results. But overall, you can see we're picking up the majority of these. I want to just now take you a bit back to classical mycology. And the reason for that is even in fungi, we can show some cute pictures. But first, I have to show you this. The soil that we streak out onto standard medium, you can't do anything with. It's completely overgrown, and there's absolutely not too much that you can do with. So we add some chemicals to specifically enrich for specific genera of fungi, and in this case, we were looking at Fusarium. From these, we isolate, and the Fusarium genus has quite a nice distribution with colors, but these usually range to the oranges, reds, and pinks. But when you look under the microscope, you can also see they have very beautiful spores and structures there. What is really good characteristic of them, on the right-hand side of the image at the bottom, you can see these banana-shaped spores. And that's a very classic Fusarium shape for that. In the middle panel, you see chlamydospores. These are survival structures. If you, for instance, have a pathogen from the Fusarium genus in soil, it can stay there for decades. And an example of this is the banana industry in the world. Fusarium oxysporum cubensi is currently decimating banana production. Even here in South Africa, we're struggling with it. And the reason for that is those chlamydospores. Once the pathogen is introduced into a region or into a farm, it stays there. You can get rid of your bananas, plant something else, because you will never get the fungus out again. Maybe I should press hard. There we go. The last thing I want to share with you is Fusarium serpentinum. This is a bit of an anomaly in the mycological world because it was described from a single species, identified here in South Africa, but we don't know from where it was. When we sampled through Tswalu from just 7 of the sites that we did look at, we found an additional 5 or 10 isolates of this. So we can now give a distribution for it. It must be semi-arid linked. Remember, we did look at the grassland biome, and through all of this, we've put about 4,500 Fusaria into the National Collection of Fungi, but we've never observed this one from any of our isolations. What makes this really a nice fungus to work with, it's not a pathogen, it's not economically of any importance, we think. But if you look at just below the culture morphology, plate E, you can see that these spores have an additional little kink in them. And this is where the name serpentinum comes from, because it was observed that they have a sort of like a cobra stance, and hence the name. This is the work done by one of our students, Maggie Manyatsa, that's in the audience. So if you want to find out a bit more about this, please speak with her on the work that she's doing. And I think then my take-home message for today is the soil biodiversity for fungi and swallow is looking really good. We can see biodiversity is very high at all of the sites that we sampled, but also across all of the distribution, we see for the species that we were looking at, we could see distribution at the majority of the sites. If we take into consideration that for every known plant species, there should be at least 8 unidentified fungal species, it means there's a lot of work still to be done. But our work shows that with 25% of the isolations or the isolates that we've identified, we can't group them into a specific genus, for instance. And then one thing that's very important to remember is that your underground biodiversity supports anything else that you have on top of that. And unfortunately, soil conservation is not the top priority when we start talking about preservation and conservation. But the ecosystems services that these microbiomes offer keeps the plants healthy. And then obviously, if you have a healthy plant and ecosystem, you will have a more healthy fauna occurring on there. Unfortunately, climate change is also affecting fungi, and that's something that we're interested in. For instance, some of the physaria that we've identified has been described about 50, 60 years ago, with absolutely no recognition of them or even finding of them anywhere else. And what happens is, about, I think, 2013, we started seeing this one being reported as a pathogen. So something must have changed since 1944, 1945, till about the early 2010s, where this is now readily observed. And incidentally, this species is also the one that we find the most in the grassland biome. So potential for new pathogen development is very high. All of this data that we've generated feeds into a bigger project that supports quarantine of import-export, because quarantine lists are based on what's occurring in a country. Unfortunately, with fungi, we don't always know, so it's very difficult to say you can't import something or export something, and these lists are helping for that. And the other thing that it presents or follows in with is that it is providing us with access of what the biodiversity is for healthy soils. And we are currently working with the Department of Agriculture to develop a high-throughput screening tool for subsistence and commercial farmers to help them with soil analyses. And with that, I would just like to thank everyone, especially the research and conservation from Oppenheimer Generations for allowing us to work on Tswalu and some of the other properties, and then also the various other instances or institutes for funding. And then lastly, I have to point out 2 collaborators. The one is Rian Pierneef, he's the bioinformatician and does a lot of the downstream data analysis, as well as Maggie Manyatsa, because you saw those images, they were really at a very high level, so thank you for that. And thank you very much then.
Thank you, Edward. We unfortunately don't have any time for questions.
Fine.
If you do have questions for Edward, please reach out to him during the lunch. Up next, we have Ara Monadjem speaking on why taxonomy and baseline biodiversity surveys still matter. Thank you.
I'd like to start by thanking the Oppenheimer Research Conference for inviting me. It's a great honor and pleasure for me to be here today. My message is very simple today. You can see from my title, it's about the fact that taxonomy baseline surveys are very important for conservation and for everything we do ecologically. I think this is a message that most of you already are familiar with, but perhaps you're not familiar with some of the details of this. And so I hope I can keep you entertained throughout this talk. We are all familiar with maps that look like this. In this particular case, it's a map of the biodiversity hotspots, the 2000 version. And these maps like this play a very important role in conservation because many of our conservation plans are based on maps like this. So where do the data come from that produce these kinds of maps? Much of the data come from museums. The Central Museum, this is the Field Museum where I did a sabbatical a few years ago. On the right, it's the Paris Museum, where I've spent many months working through the rodent collection. On the left are two museums in Germany, Berlin and Bonn, which have very important collections from Africa. If you want to study African small mammals, you will need to be familiar with museums overseas in Europe and North America, because that's where most of our collections are. And where do the data come from? Where do those specimens come from? They come from field surveys conducted traditionally and in the past almost entirely by European or European-led research teams. This is one of the expeditions I was on in Liberia in 2005 during the dry season, where it rained on us every single night. So here's a message I've taken from a paper that was published in 1954, Waldo Meester. Many of you may have heard that name. And if I can read it, Africa is still in the exploration and cataloging stage as far as mammalogy is concerned. My message for today is that 75 years later, we are still in that same situation. And here's a paper that was published last year by Harith Farouk, a Mozambican colleague and friend of mine, that shows this actually very well, and I could actually end my talk with this slide. On the left and on the right are herps and birds, so we can ignore those. Actually, they show exactly the same trends. Let's look at what he showed for mammals. He was trying to answer the question, how well surveyed is Africa for vertebrates? The top map shows all the squares in different colors, in red and blue, that have been surveyed in the past. Each square is approximately 100 by 100 kilometers, and you can see looks like there's good coverage across Africa until you realize that the red squares represent only one or at most two surveys in that 100 by 100 square area. So what he did is he took the data there from those maps and he corrected for the size of the country, etc., to produce a map below to show how well surveyed is each African country, in this case for mammals. And if the color is yellow or brown, it means you basically have not been surveyed at all. And you can see that there are only two countries, Benin and Rwanda, that show up blue, which is relatively well surveyed for the size of the country. So why are we not investing in field surveys? One of the reasons is maybe not that obvious, but these field surveys are tough. You've got to work in sweaty, hot conditions. That was my mammalogy lab on the left, where you can just make out the sleeping quarters behind, where there were 50 people sleeping, trying to sleep while I was doing work on bats. On the right, that was a trip a few years ago to Angola. I didn't even have a tent there. I had to sleep out in the open. You struggle to find water and food, and you struggle to find means of disposing of said water and food. So it could be that we don't want to do these field trips. They're just too tough. But there's more important reasons as well. As a scientist employed to do science, the kind of work that the sort of publications you produce with field surveys, such as this RAP Bulletin of Biological Assessment, and I've published many such papers, they don't count for anything as a scientist. You can't even put them in your promotion package, and they are not rated at all. And I was told from a long time ago, stop, as an ecologist, because I'm not a taxonomist, I'm an ecologist. My ecologist colleagues told me, stop doing these surveys and start publishing proper ecological papers. And it's not that I don't agree with them, but this work, somebody has to do this work. So taxonomically orientated papers are not recognized by the scientific field. There's less and less money for this kind of work. If I were to say community engagement plus this plus that, you can get millions, but if you say to go and survey for biodiversity, you get nothing. And then there are all these more and more regulations coming out which are not preventing poachers and illegal harvesters from taking what they want, but they're regulating more and more the zoologists that need to do the work. So here's a map of, and I produced this just for this talk, to show you all the expeditions I've gone on for specifically small mammal surveys, where I've spent, most of these sites I've spent at least 1 month surveying them. Many of them I've repeatedly visited them, over 50 surveys in total. And you can see literally I have surveyed my home country of Eswatini off the map entirely. And these are the animals that I study. Bats. I also work on rodents and shrews, but for this talk I want to focus on the bats. All the photos you see in this talk are my own. If I could take a decent photograph, imagine how beautiful these animals would really look. They are absolutely fantastic creatures to work with. And what is more important is that we're discovering new species at an accelerated rate in the in the last 2 decades. The black line shows the accumulation of species total of bats that we know from Africa. But if you look at the bars below, you can see that for most of the 20th century, there were hardly any new species being described. But in the last 2 decades, those bars have started to increase quite radically. So in practice, what does this look like? This is the sites I've surveyed in West Africa. And in particular, what you need to know is that the West African forests fall under the Upper Guinea rainforest zone. It's a biodiversity hotspot. But I want to focus on the borderlands between Liberia and Guinea. These are upland areas. Most of West Africa is flat and lowland, but here we've got some low mountains, high hills, low mountains with incredible diversity. And stunning landscapes. There is rainforest, there is grassland mountains, clear flowing streams. It's one of the true paradises of this world, but most of you may have perhaps never heard of this area. And what have we discovered here? From surveys that I have done, 1 new species, 2nd new species, 3rd, 4 new species based on a single survey from 1 family of bats, the Vespertilionid bats. These are the equivalents of the passerines when we talk about birds. On the right, I'm showing a phylogeny only for you to see that these 4 species have very deep genetic divergence. It's not a matter of, oh, let's split this from another one that's close. It's not like that. There are 10 species of Vespa bats all living together in that same zone and all easily identifiable both in the hand as well as genetically. And then a 5th species from a different family, the Miniopteridae, the bent-wing bats, bringing the total to 5 new species from a single, well, it was actually 2 different surveys, but from a single site. And you might think, okay, this is an area far away in the deep jungles of Africa that hasn't been explored, but it's not the case. Mount Nimba, where this work was done, was very well surveyed in the 1960s and the 1970s, as well as in the 1980s, for bats. And to the extent that André Brosset in 1984 could write this, for those of you that don't read French, it says, the bats of Mount Nimba are among the best studied in Africa. And it was a true statement. Imagine what we're going to find in areas that have not been well surveyed. Okay, so we found a whole lot of new species. So what? Well, there are conservation implications for this. This is the lowland tropical rainforest zone of Africa, the Congo Basin, to the west, the Lower Guinea rainforests, and then the Upper Guinea rainforests of West Africa. The Upper Guinea rainforest, as I mentioned before, is a biodiversity hotspot. It spans a broad area, approximately 1,600 kilometers. So how do you conserve such a large area? Compare this to the upland zone where I am focusing on here, where the sites that I've surveyed, or I've been part of teams that have surveyed, are barely 100 kilometers apart. In the Upper Guinea rainforest, there are 12 species of bats that are endemic to the Upper Guinea. Of those 12, 9 of them are endemic to the upland zone of Upper Guinea. I don't even know what to call it because it doesn't have a name. It hasn't been recognized as a bioregion or as any other kind of region. But here's an area that's hyper-diverse, not just for bats. It's also diverse for reptiles. It's also diverse for frogs, for plants. But it has been overlooked in part because it hadn't been studied properly. And here's to show those 9 endemic species from this upland zone. 6 of those species were described since the year 2000, and 5 of them based on the surveys that I was involved in. So this, I hope, shows you the importance of continuing doing baseline survey work with correct taxonomic work alongside it. I want to show you a slightly different angle now to this whole story, and that is what I've called here the disappearance of Hippocydrus lamotti. This is a bat that was described from Mount Nimba, absolutely gorgeous part of the world that has vanished. It was described in 1984. It was never seen again. The site at which it was described is in a mining zone on the Guinea side of the mountain, and the mining company employed an EIA that employed a bat expert that the sole purpose was to tell them, does this bat occur within the mining area? The bat expert couldn't make any sense of it and hired me to come and tell them, does Hippocydrus lamotti actually occur on this mountain where they're going to mine? So I did a survey there and found 7 species of the family Hippocydrus, the leaf-nosed bats, 6 of them clearly defined, and we know what they are, even though the two at the top don't have actual names yet. But then there was one species clearly distinct morphologically and very clearly distinct genetically, which we presumed could be Hipposideros lamotti, but its measurements did not agree with the measurements of the description of the species. The description is in the middle column. There's measurements. If you look at the bar in the red, you can see there's big discrepancy between the measurements of our specimens compared to the measurements of the type description in the length of the fingers of this bat. To answer this question, I had to go to the Paris Museum, stunning old 19th century building, 18th century old building with beautiful gardens, but that's not where the mammal collection is. The mammal collection is on 55 Rue de Buffon on the side, and it looks like it was taken straight after the World War, this photograph, but I took this quite recently. Thankfully, the building is being renovated now, so hopefully it will look better in the near future. And this is where the type specimen of Hipposideros lamotti is. That's its type, that's its specimen number. And when I measured this, I remeasured the type specimen and look at the discrepancy between my measurements and the original measurements. The remeasuring of the specimen allowed me to make the conclusion that the original describer of the species had measured this animal, and that indeed we do have Hipposideros lamotti on that mountain. That single statement in that report unlocked hundreds of thousands of dollars for mitigation for bat conservation. So taxonomy really can be vitally important for conservation. What about ecology, biogeography? I want to move away now a bit from that to see what else can we learn from having good survey data and doing good taxonomy. I want to focus on the montane regions of Africa for this next section, and they can be roughly divided into these mountain regions, at least from the perspective of mammals, although many other groups will follow the same line here. And if we take all of these areas in red, and we look at what species of bats occur in these areas, and then try to compare them in terms of, from a species composition point of view, how do these sites compare? I can do an NMDS analysis like this, which will mean nothing to anybody. So I will take this and I'll map it back onto that original map. What I want to just mention here is each of these circles is one of those mountain zones, and you can see clearly 3 mountain zones are spread away. If the dots are close together, it means species composition is similar. If it's far away, it means they're different. Let's map this back onto the map of African highlands, and you can see that there's a whole lot of areas in gray that share similar species composition, but the Drakensberg area is quite unique and different from it. And quite amazingly, the Albertine Rift and the Cameroon volcanic line are very similar and very different from the East African mountain systems. So in terms of species composition, the Albertine Rift shares affinities with the Cameroon volcanic line rather than with the adjacent East African mountains. What about the Angolan highlands? Until a few years ago, we had no data on this, but in 2022, I was on an expedition there where we worked the Kumbira forest and the Namba mountains and the Namib forest. It's an absolutely stunning area of the world. The Namib forest, you can just see it on the far left of the map, and this is the largest remaining Afromontane forest in this Angolan escarpment. And in that short survey of 2 weeks, there are 5 new species to science. There they are over there. And what can they tell us about biogeographical relationships? Well, of those 5 species, the 2 Rhinolophus species, the 2 horseshoe bats, share affinities with further south, with South Africa, Namibia. The Hipposiderus, the leaf-nosed bat, shares affinities with the Congo Basin. And the Miniopterus shares affinities with the East African highlands. These are all new species to science, but their closest relatives come from very different parts. So to understand where this unique Western Angolan highlands community comes from, we need this kind of taxonomic information. Then I'd like to move on to the other side now and look at a new bioregion that we described that was led by Julian Bayliss, the Siama, or the Southeast African Montane Archipelago. These are a series of low mountains. In fact, they're probably just nice hills. But on the left, you can see that's Mount Mabu, blanketed in rainforest, medium elevation rainforest, 7,000 hectares of it, which is the biggest rainforest that we have in Southern Africa, if you include Mount Mabu in Southern Africa. But around all of these mountains is lowland hot savannas with baobabs dominating the landscape. So it's not surprising that the small mammals in these Afromontane zones are strikingly different from those in the savannas surrounding the mountains. It's not even that there's differences in species. At a genus level, the community of rodents is completely different in the forest compared to the savannas surrounding them. This is what it looks like if we were to survey, which we have done, the small mammals in the savannas around Mabu and the Mount Mabu itself. There are 6 species of rodents that live on the mountain. There's actually more than 6 species that live in the savannas, but 6 that are right on the edge of the forest in the savannas, and none that cross that forest-savanna divide. But when we look at the bats, that's not the case. There are 9 species of bats that occur all the way up this mountain, from savanna into rainforest. And so you might say, okay, bats, they have wings, they can fly, and therefore they ignore habitat differences. But maybe this is not the case. There are 2 species of bats here. If you look under the montane, you see there's 2 species that are restricted to the montane. One of those 2 species is a Miniopterus. And if we had done this analysis before the taxonomic work I did on this, it would not appear under montane, because until very recently, we thought there was only 1 species of Miniopterus in Mozambique, Miniopterus natalensis, that occurs across the whole region. But based on survey work that I've done in Mozambique over the years, we showed that there's a lowland species, Miniopterus mozambicus, that occurs very widespread in the lowlands of Mozambique, and there's a very different species, Miniopterus wilsoni, which we named after E.O. Wilson, that in fact put a lot of effort and raised a lot of money for Mount Gorongosa, where this bat was described from. And therefore, the high elevation Miniopterus is, we have shown, is a different species from the lowlands. But this was not known before we did this work. And all of those would have been lumped together. In fact, we've shown that there's 2 other species in Mozambique. So instead of Mozambique having a single species of Miniopterus, there are 4 different species, all very easy to identify in the field, all clearly genetically distinct. So again, this is not about splitting and saying, oh no, I think this way or that way or whatever. So even for ecology and biogeography, such taxonomic work, it becomes important. I'd like to just end off to say, if you're interested in small mammals, please look up some of my books and help me raise more money for my work. In the last 15 years, I've been paid, I think, 5,000 Rand because of the sales of my books. That's, I'm not so good at maths. What is that? 5,000 divided by 15 years, it doesn't come out to very much per year. I'd like to also say a big thank you. I have so many people to thank that it's very difficult to do this meaningfully because every expedition has been with different people. But I'd really like to thank the students that I've worked with, my own students from my lab, my master's, PhD students, and ex-master's, PhD students that have joined me in the field and themselves have become excellent field biologists. And finally, I would really like to say a big thank you to the small mammals themselves for having brought so much joy and happiness to my life over the years. That's a pygmy mouse. Thank you.
Thank you, Ara. Do we have any questions for Ara? Oh, we have one at the back there. Sorry.
Hi. Yeah, it's not so much a question, but it's relevant because, Ara, your presentation may end up being one of the most important ones of this conference. Field work is flippin' hard. And I sense, well, in my experience, I'm relating this now to the farming community around the Eastern Karoo where I work. There are no youngsters coming in because work is, it's hard and it's much easier on TikTok and it smells very different in person. So it's kind of an appeal. There will always be funding for community work because communities vote in elections. If we don't fund this kind of stuff here, to get this field work on, keep it ongoing, there's going to be nothing for our scientists to look back at in 20 or 30 or 40 years' time. So from that point, thank you, and thank you for all those adorable, cute little flying mouses that you found.
Thank you. I'd just like to just add to that. We've had some brilliant talks this morning, and the talk by Belinda Reyers, she mentioned the disconnect between humans and biodiversity. And I strongly believe that the direction our biodiversity laws are going is that they are helping to create that disconnect. And we don't need it. We need more connection with nature, not less. And the more disconnected we come, the more we are told, don't touch and stay away, the less likely we are to appreciate it. We all love animals, and we should be allowed to love those animals and ensure that that's the best way to make sure that they remain on this planet.
Thank you so much, Ara. Round of applause, Ara. And our final talk for the session is Gareth Hempson speaking on seasonal variation and drivers of parasite prevalence in migratory Serengeti wildebeest.
Okay. Cool. Hello, everyone. Just to start off with, firstly, a very big thank you to the Oppenheimer family for the opportunity to be here. It's my first time and really appreciate it. It's lovely to be here. Secondly, just to say the work that I'm presenting represents the efforts of a whole big crew of co-authors and collaborators, and everyone's played really important roles in producing this work. Okay, so today I'm going to take us to the Serengeti in Tanzania, and I'm going to be talking about wildebeest and their parasites. Okay, so one of the key features of the Serengeti wildebeest population is their movement. So they're renowned for the migration that they undertake, and it's really the sort of standout feature of the population. But why do animals move? So one obvious important reason is to access forage. Okay. So these are the shortgrass plains, and that's where the wildebeest spend most of the wet season. There's a lot of high-quality forage available there. But in the dry season, this forage doesn't regrow, and there's not enough food to support the animals. So they all migrate up to the north of the system, to the Mara region, and in these areas there's reserve forage that then sustains the population. There's also drinking water, which is critical to the population persistence. Another good reason to move is to escape predators. So predators have territories, their young are usually quite weak and they can't follow you, so by moving you can often get away from predators. And then something that we see in the Serengeti then is that the shortgrass plains themselves are essentially predator-free zones because, well, low predation, because there's just no prey-based sustained predators out there during the dry season. So now you've got lots of food, so you're able to smooth your food resource through the year by migrating. You're escaping predation to some degree. And the consequence of that is that you're able to build up these very high population sizes. So with high population sizes, you get dense aggregations of animals, and these are conditions that are ripe for pathogen spread and disease spread. And so this is where we enter the story, and we're starting to ask, well, what's going on with parasites in this population? You know, you've got this really abundant population. How are they shaping the ecology of Serengeti wildebeest? Okay, so for the work I've done, the stars of the show are these GPS-collared wildebeest. I've had the privilege of spending a large chunk of 2 years out in the field in Serengeti. I was there from October through to December, 22 to 24 there. Worked on 54 different GPS-collared wildebeest over that time. So those GPS collars provide us with a wealth of data. Obviously, they tell us, they give us information on animal movement, so we know how fast they're moving, where they're moving, so those real sort of patterns of movement. But they also provide, you know, those locations that those animals are using. So we can use those locations to understand, to characterize the habitats that they use. And then from that, we can then use, say, remote sensing products to understand something about tree cover or rainfall or fire. And so we can extract those variables that characterize habitats. Another important part of the work I was doing in the field was to go and try and catch up with all of the animals, the collared animals, every 2 weeks to 2 months. And so when I could catch up with them, the main purpose was to collect a dung sample from that individual. So we're building this time series of dung samples from individuals. And the main point of those dung samples was so that we could get them back to the lab, where they would count the parasite eggs. These are strong-gile nematodes. Count those eggs in the dung. So that gives us a sense of their parasite burden. Then another important thing I did while I was there was to record whether the animal had a calf or not. Okay, so that's a quick overview of the data that are going to appear in this talk. The next thing to say then is to give you a bit of a background on these gastrointestinal nematodes that I've been studying. And so when I talk about parasites today, I'm talking about these nematodes. So hopefully you can see just from the figure there, just to give you a sense of their life cycle. You've got an adult worm that's in the gut of the wildebeest in this case. It's producing eggs. Those eggs are released in the dung. They hatch in the dung. They go through about 3 larval stages in the grass layer. And the L3s, the third life stage, then crawl up the grass, and they get eaten by the wildebeest, and then they mature to adults, and the cycle continues. Okay, so there's a free living stage and an internal stage. Okay. Whoops, this one. Don't be scared. Okay, so this leads us to the point then to ask, like, what drives variation in these parasite burdens in Serengeti wildebeest? Okay, so that's our first question that we're going to tackle. So we've got those fecal egg counts, but it turns out that they're a very noisy approximation of what the actual worm burden is inside the adult wildebeest. Okay? So to that end, you've had a preview of this, we put together a state space model. So this model has been developed largely by Juan Morales, a colleague of mine, and I'm going to walk you through it. This kind of thing usually scares me. I'm getting the hang of this one. And I think something important to note here is that there's that top part, that hidden process, those are all things that are going on inside the wildebeest. These are things we cannot observe. And so those are things we're modeling. And then the data that we've got at the bottom there, you see there's an FEC. That's the fecal egg count. These are from the dung samples. Okay, so that's an observed process at the bottom. Are we moving on? Right, and so to start breaking this down a bit, there's this top component, that first equation, that tells us something about the ingestion of larvae by the wildebeest. The next part is looking at the maturation of those larvae inside the wildebeest to become adult worms. Then we look at the population dynamics of those adult worms, like what determines how many they are. And then the last part looks at their fecundity, so how many eggs they're producing. And that would then link to those fecal egg counts that we make. Okay, so I'm going to walk through the model in more detail and show you the results at the same time. So now we're looking at this larval ingestion by the wildebeest. So the first thing to think about is how much dung is landing in that landscape, because the eggs are in the dung, so we should think that if you've got more wildebeest in an area, more dung, more eggs on average, and then if you go forage in that area, there should be more larvae. Model suggests that there's a slight positive effect of wildebeest densities on larval ingestion. It's not convincingly different from zero. Different formulations of the model, you get stronger, weaker effects. But it's not a strong, clear effect at this point. The next part then is that, so these larvae are in the grass layer, they've hatched, and now they need to actually survive there. And so the better they survive, the more you're likely to consume. And so the microclimate is important, so things that matter are things like tree cover, which provides shade, keeps it cooler. Rainfall keeps conditions moist, which promotes larval survival. And then something like fire kills larvae. The estimates our model is producing suggest that there's no effect of tree cover, no effect of fire, and rainfall, similarly, nothing convincing. In the right direction, but not a convincing effect of rainfall. I just want to add, though, that all of these things, we've studied them experimentally at local scale, and we have seen clear effects of each of these variables, but not coming out in the model we're fitting at the moment. Okay. The next part that's going to influence how many larvae you ingest is your appetite. And something that has a big effect on your appetite is your reproductive status. So in the last stages of pregnancy and during peak lactation, the energetic demands of female wildebeest is much higher. So they need to eat more food to meet those demands, and they're likely to ingest more larvae. And in this model, we see a very strong support for that kind of effect. Our adult females with calves would seem to eat a lot more and therefore ingest many more of these L3 larvae. Okay. The next stage, this is a bit of housekeeping. It allows some maturation time for the larvae to become adults. And then the next one is just looking at the effect of this maturation time from larvae to adults. And so what we've done here, oh no, sorry, I'm getting distracted. So we're looking at this adult worm population. There's two components. The inputs, so that's how many larvae are maturing to become adults, and then the survival, so how many of those are actually dying over time. We've interacted both of these effects with your calf burden. And the reason whether you've got a calf or not, The reason for that is that those energetic demands also suppress your immune system. So if you have got a calf, we'd expect you for more larvae to mature and fewer adults to die. Our model picks up no support for that, but on the next stage, there's suggestion of a very clear immunosuppression effect on that adult worm fecundity. So animals with calves, the worms inside them, the model suggests that they're producing a lot more eggs. Okay. So with all of that done, we can then go to an individual. The red dots are the actual fecal egg counts from the dung samples I collected. The dark line is the estimate of the internal worm burden. It's doing a reasonable job. It's a nice example. For this animal, it didn't have a calf in the first year of the study. It did have in the second. So you can see these clear effects of having a calf on the fecal egg counts and the worm burden. Okay. So how do these parasite burdens influence wildebeest movement and habitat selection? To address this part of the study, we used integrated step selection analyses. So for each step in an animal's movement trajectory, you generate random points that they didn't take, and you compare those to the steps that they did take. This form of analysis allows us to take account for movement constraints when assessing habitat selection, and vice versa. I should be fitting this model using the estimate of the worm burden. I'm using the calf effect in the meantime. So time is running here. But I just want to say that our worm burden results in the preliminary models we fit align with the calf effects, but they're not quite as clear. So in this model, you've got a movement component that you're analyzing, interacted with having a calf, and a habitat selection component interacted with having a calf. And to jump straight to the results, what we see is that there are quite strong effects of having a calf on your movement. Basically, if you've got a calf, you take shorter steps, so you don't move as far in each time step. This is a 12-hour time step, and your turning angle tends to be greater. So the net effect of this is that animals with calves tend to show less displacement over time for each time step. Okay. Not much going on the habitat selection side, and I'm going to skip over it because it's not something important for the take-home messages for today, which we will get to. There we go. Right, okay, so to wrap this all up, how do these parasites influence the Serengeti wildebeest population? So I think what we're seeing here is that you're getting the wet season really is this crunch time. You're getting these big aggregations of calving females, and because there's lots of animals, there's lots of dung, so you're getting lots of eggs going into the system. Their appetite is enhanced. So they've got huge energetic demands, so they're eating more, so more larvae are being consumed. That the costs of reproduction suppress their immune system, not influencing that larval development, but we saw that effect on the number of eggs produced. So there's more worm eggs going into the environment. And then we're also seeing that these effects on animal movement, that animals with calves are less likely to move out of that area. So it sort of creates this cycle that just feeds back on itself. So how do you escape this? Well, you move. So when those calves are big enough and they're able to move off and the system dries out, then movement allows you this migratory escape from parasites in the landscape. So in addition to moving to access food and to escape predators, movement also allows you to escape parasite burdens. All right, I'll leave it there. Thank you very much.
Thank you, Gareth. We can take one quick question. We have one towards the back. Is there a mic? Towards the back. Yeah.
Hello, and thank you for such an insightful presentation. My name is Lydia from Stellenbosch University with the African Wildlife Economy Institute. And I just wanted to ask a question on, you mentioned something around the parasites affecting the body condition, and I wanted to find out from you if this means that there's any implication for disease transmission potential for other zoonotic diseases in these herds?
Struggling here a little bit, but so are parasites having, like, if they leading to declines in body condition, that these could have knock-on effects for other zoonotic diseases? Was that your question?
Yes, so I wanted to find out if since it affects their body condition, does that mean that there's any implications for disease transmission for other zoonotic diseases within these herds? Like, have you noticed any other potential or implications around that?
We haven't looked at other things specifically. I'd imagine that those effects would exist. Something that's perhaps quite a jump is just we've been looking at the implications of flushing livestock with parasites that are not resistant to anthelmintics. So we've been studying the livestock populations around the edge of the park where the animals have been treated with anthelmintics, developing resistance. And then you get all these naive parasites brought in as the wildebeest migrate through. So yeah, it's not really linking to your reduced body condition and disease, but there's certainly lots of implications for all this movement, disease spread, shifts in body condition. Yeah, lots of knock-on effects through the system, yeah.
Okay,
all
thank
right.
you. Thanks, Gareth.
Cool, pleasure. Thank you.
Just before we end off, can we give one final round for our exceptional presenters during this session? A few housekeeping rules. Before we head off to lunch, we're going to take the conference photograph. That'll be taken by the pavilion. All those who've attended in previous years will know where to go. And those who don't know where to go, please follow Duncan McFadden. He's going to raise his hand here. I think everybody knows who Duncan is. And he'll lead you to the pavilion. After the pavilion, you guys can all head off to lunch. Enjoy your lunch and be back by 2 p.m., please. Thank you, everyone.